Um, I actually didn't erase the stuff I was afraid I did, so I do have it here. If people didn't get it, but we do need to go on, so it's sitting here on the screen while I'm recording, so you can go to the video to get all of this if you need to. Question?
Before we start, I just have a question.
Mm-hmm.
So one of the questions is on the thing on the bottom, the makeup one we were looking at as we were doing it together, and we cannot figure out what the unmyelated
axons.
All I could find was like tickling and aching pain.
That's pretty much right.
Like what is?
I think it asks like where they were and what they were
for.
Like, can you maybe explain it a little?
Well, the point of the question is not really just for you to find an answer somewhere else, but to think about what myelination does and when it's not necessary.
So answer kind of that way.
So if you answer it with a thoughtful response as to why it's not in certain types of tissue, or I mean why it's not wrapped around all axons. You can look up and find out some examples of ones that aren't myelinated, and that might give you a clue as to thinking along those lines, but you can go a little bit further than that too. So just explain why axons might not need to be myelinated.
Okay.
That's essentially what the question is getting at. So,
thank you.
Yeah. Okay. So over the break, I did a couple of things. One, I changed the due date for the lab quiz that's in this week's folder. It's now due, what did I say? That was May 3rd, because we won't get to that stuff today. We won't get to it until next week. Some of what that quiz is on is the micrographs we looked at last week, but there's more about the anatomy that's on the quiz. So I pushed that due date back so you'll get that information before you have to complete it. Also, I went ahead and changed the pointers for current week and next week, so that's updated. And I put a link in for this video. If you go into the video content section in Blackboard, I just put the link in, so it's the last one on the page. Okay. So if you wanted to watch that again, here it is. It's embedded here, and when they're embedded, you can't resize the window. You might be able to do that through player controls, or what you can do is, notice as I hold the mouse over the title in the video here, if you click on that, it'll take you to the YouTube page, and you can play it there and enlarge the screen as you see fit. You can also do a search for the title of the video. Yeah, just a Google search, and it'll take you to the YouTube link to it. So if you want to see it in larger, you can. Yeah, the player controls don't do anything special for you. So you can go to the YouTube page itself like that, and you can see it there.
How does the pain?
But we've already done that. So just if you want to watch that again, at some point I need to reorder these. So if you're looking, actually I'm going to do that right now. I don't want it at the end of this because there's a few videos that have to do with next week's stuff and some other stuff. So actually I put it here now. It's the first of the nervous system videos, but it's in that section there. So to continue on, The next thing I want to talk about, which actually takes us back to YouTube, is the propagation of the action potential. There's another video that I have in my history here that's showing how the action potential propagates. Now, before I show you this, let me just talk a little bit about what the point of this is. This is a very brief video. It's only 48 seconds long. But the word propagation, in this sense, is talking about how the action potential moves down the axon. The word propagation is related to the word propaganda. Hopefully you can see that they're very similar words. Propaganda is referring to information that's moving. If you're trying to get propaganda out, you're trying to get a particular message out. It usually has a negative connotation, which isn't terribly important, but that's what it's talking about, getting information out. So propagation here is talking about the information that is the electrical signal moving down the axon. So that's what this video is about. Let me play it real quick, and then I'll talk about it.
An action potential, depicted as a red band, is propagated in one direction along the axon. During an action potential, the inside of the cell membrane becomes positive with respect to the outside. An action potential generates local currents that tend to depolarize the membrane immediately adjacent to the action potential. When depolarization caused by the local currents reaches threshold, a new action potential is produced adjacent to the original one. Action potential propagation occurs in one direction because the recently depolarized area of the membrane is in absolute refractory period and cannot generate an action potential.
Okay. Why does it keep doing this to me? Gotta change it. So, I like this.
Adjacent to the action potential.
I like this video because it does a good job of showing the action potential represented as where the red band is. This is where that change in membrane potential is taking place. The movement of the positive and negative charges sets up what we can consider a current. Again, that's kind of a metaphor from electrical physics. But it's also kind of really what's happening with the ions. The ions are moving into the membrane here. Sodium ions are moving in. And so more sodium ions are going to move in to sort of the space left over when those first ions moved in. And so that charge, that positive charge, is moving in, and it's going to continue to move in to the cell. And as those ions move in, they'll diffuse in either direction. They'll go down the axon this way, and they'll go up the axon this way from when they come in. Now, at this point in the voiceover, they mention that the action potential only moves in one direction because the segment of the axon that was just in the going through the action potential is in what's called, I guess they never actually labeled it. So here's the action potential taking place. Where it just was a second ago, what's represented by the yellow band here, is representing what's called the refractory period. So let me talk about that for a second. This is when a new action potential cannot be started by a new stimulus. Now, there's actually two phases to the refractory period, and what they're talking about in the video there is the absolute refractory period. Which means absolutely no new action potentials. And this is really because the sodium, the voltage-gated sodium channels are inactive. Okay, I was talking about the two gates, the one that opens at -55 and the one that closes at +30. When that second gate is closed, when it hits +30, we call that channel inactive. It's in fact called the inactivation gate. And so sodium can't move through it at that point. So if sodium can't move across the membrane, we're not going to get another action potential. Absolutely. We have to have the sodium. Without it, nothing happens. The other aspect of refractory period is called the relative refractory period, which means a stronger stimulus is needed to start a new action potential. So as I'm defining a refractory period up here, I say when a new action potential cannot be started by a new stimulus. What's implied there is that it's the same stimulus. So if we've got from resting potential to threshold by depolarizing 15 millivolts, depolarizing 15 millivolts isn't going to do it during the relative refractory period. We need to depolarize a lot more than that. Okay, and the reason for this is the stimulus has to overcome the potassium leaving the cell. So let me go back to the graph here. Really, we could say the absolute refractory period is when we're in depolarization and the start of repolarization. During depolarization, all sodium channels are open, so we can't get more sodium in. Nothing's going to happen. Nothing more is going to happen than what's going on in depolarization. But once we hit +30, the sodium channels get inactivated, and they'll stay inactivated until we get past threshold on the other side. Okay? Threshold is when the first gate opened and allowed the sodium through. At the peak is when the second gate closes. The second gate stays closed until the first gate closes back again. So we go up past -55, the channel opens. We go back past -55, the inactivated gate stays closed, and the first gate closes also. So everything's closed. Then the inactivation gate can open again. But once we're down here, -15 millivolt depolarization is not going to get us back up to threshold. Even if we're right here and we're like 2 millivolts away from threshold, as we depolarize, that depolarization is just going to slow down the hyperpolarization. It might slow it down and get us to resting membrane potential directly and not ever go past it. But to get to threshold, we have to not only overcome hyperpolarization, but reverse it and get back up to the threshold. So a stronger stimulus is needed to overcome what's happening with the potassium. So back to the video here. What they're saying at this point is sodium's rushing into the axon at this point, and it diffuses in both directions. But when it diffuses back this way, it doesn't help this part of the axons in the absolute refractory period. So depolarizing is not going to cause another action potential, which is good because we don't want the action potential to go the other way. And then as it moves down further, sodium will still diffuse back to where it just was going through the action potential. But in this part here, we're in the absolute refractory period again, so nothing's going to happen there, and it's only going to move forward. Oops.
Refractory period.
Sorry.
And cannot with respect to the outside.
Why is it doing that to me? Okay. So back here, just to show it with the whole axon. Okay, the red. Oh, that's right. The red thing. Oh, I didn't grab the right place. Okay. So the red thing's moving along the axon. That's propagation. And if you look closely, the way this is animated, it's sort of jumping from a section right next to the next section. And it's not going back the other way because of that refractory period. It's not allowing it to travel back the other way. Okay. Now, you can actually get an action potential to go the wrong direction, but it's very artificial. What you'd have to do is to apply an electrical shock to the axon in the middle, and from that point, the action potential will go in either direction. But that's not going to happen in a regular situation, because we don't have strong electrical signals arriving in the middle of the axon. They're always arriving at the dendrite or cell body region, so they can only go down the axon in one direction. Yeah.
If you do like electrical muscle stimulation and things like that, would that trigger those reverse action potentials?
Yeah. So if we took an electrical wire and put it in here and stimulate this axon right here, it would cause an action potential to go from here to the end of the axon, but also because the adjacent membrane, which is upstream, if you will, from the point of stimulation, since it's not in a refractory period, then the electrical signal will also cause an action potential to travel that way. When it goes back to the original cell body, nothing really happens.
Yeah.
Okay. But yeah, if you do artificially stimulate an axon in the middle, action potentials will travel in both ways.
Yeah.
Yeah.
There's no like negative side effects that come of that, right? Like,
It's possible that overstimulating particular neurons can cause damage, yeah, but I'm not familiar with the data on what happens there. I just know that one way to lesion tissue is to just shock it with electrical wire.
yeah, and if you like do a constant DC voltage or something, it'll make your blood turn into or make basically have your blood turn into lye.
Turn into
Yeah,
lye, did you say?
yeah.
I'm not familiar enough with what's going on, but I could imagine, yeah, maybe they're make it very basic or something like that. But more what I'm talking about is if you electrically stimulate neurons too much, they'll die. Yes. If you're doing electrical stimulation for some practical purpose, somebody with Parkinson's disease or something like that, you might not be applying enough electrical current to do any damage. But I'm not familiar with that data. So, but anyways, so this is a very brief little video just to show the idea that the action potential is happening at a specific place on the membrane, and then it spreads down the membrane, which is only happening in the axon for that to happen. That's the basic idea of propagation. Now, this is an unmyelinated axon. Okay. So what we're talking about here is the basic idea of propagation, but when we add myelination into it, it changes a little bit. So before I go to myelination, any questions here? Okay. So before I leave the video, let's just talk about myelination in this context. So if I had myelin around this axon, let's say it's wrapped around this part of the axon, the membrane does not have the ability for ions to diffuse across it when it's wrapped with myelin. Okay. So ions can diffuse through here, but they can't diffuse through here if this is where the myelin is wrapped around. Okay. What we're talking about, let me see if I can get to this particular thing. So what we're talking about here, where the positive charge is coming in, is diffusing either direction. If there's myelin wrapped around the axon at this point, when the sodium comes in and diffuses this way, it can't just come right back out. It has to keep going, diffusing down this direction. And because myelin's there, nothing can diffuse out of the cell at that point. It makes it so that the sodium can diffuse kind of as far as it possibly can. Okay, so let me see. So let me, I've not watched this video before, but let me pull it up. It's only 30 seconds long. And I muted it.
The improved efficiency is due to the fact that the insulation provided by certain cells, called Schwann cells, which have wrapped themselves around each axon, effectively limits the movement of ions to short segments between these cells, rather than allowing this ion exchange to take place along the entire length of the nerve fiber. Loss of the myelin sheath can cause serious impairment of function of the nerves involved.
That was kind of brief. Let's see what this one says.
Let's learn about saltatory conduction. Saltatory conduction is a process that occurs in neurons with myelinated axons. Myelin is what makes white matter white. Remember, the parts of the neuron include the cell body, dendrites, axon, and axon terminal. Myelinated axons have Schwann cells that produce myelin, a lipid substance. They also have gaps in the myelin called the nodes of Ranvier. Let's take a closer look at the myelin sheath. Here is an electron microscope picture of an actual myelinated axon. We can see the nucleus of the Schwann cell, the cell itself, and the axon on the inside. Notice how myelin wraps around the axon. The gaps in the myelin sheath are called nodes of Ranvier. They play an important role in saltatory conduction. Let's take a closer look at a node. The nodes contain a couple of important transport proteins. These are voltage-gated sodium channels and voltage-gated potassium channels. Here's how it works. We will begin with all of the channels closed. Let's zoom in to take a closer look. There is a large sodium gradient with more sodium outside of the cell than in. When an action potential propagates down the axon, it reaches a node. The sodium gates open in response to the action potential. Sodium rushes into the axon by moving down its gradient. This causes the area to depolarize. The sodium gates then close. There is also a potassium gradient with more potassium inside of the cell than out.
Once the voltage reaches about positive 30 millivolts, the potassium gates take over, and potassium moves out of the axon. This causes the area to repolarize for the next action potential. If you were to observe this mechanism, it would appear that the action potential would jump from node to node. Myelinated axons conduct action potentials much faster than unmyelinated axons. We hope you've learned something about saltatory conduction, and see you next time.
Okay, so
Let's learn about
that was kind of an interesting little video for my sake. I've not seen this Go animate thing, but that's kind of interesting. Let's see. Okay, now I want to go back to the previous video first. So this, I went on to the second video because I thought this really seemed to be a fragment of a larger thing. But what we're seeing here is that's the node, that's exposed axon with myelin sheaths on either side of it. The sodium that's diffusing within the axon that we were just talking about will diffuse along inside, not allowing to escape because of the myelin until it gets here. As it's diffusing, the voltage at the membrane is going to be slowly falling off. But as long as the voltage, once it gets here, is still at threshold, that'll open up those sodium channels that the second video was showing. And sodium will rush in here, and we get an action potential there. The sodium that comes in can then diffuse down the myelin to the next node. It'll also diffuse back along the myelin to the previous node, but the previous node is in the refractory period, so nothing's going to happen there. I kind of like the way this is animated. So these white lines are kind of jumping across, and then we see a little flash going along that little part of the axon at the node. Okay, and we see the positive negative switch, which is the action potentials happening there. And then, later on in the video, they're saying something like loss of myelination can cause problems. So here we have an axon that now has no myelination, and so the action potential has to continually travel along. It doesn't happen right here and then jump down and happen over here. Okay, that's what myelin does. It speeds things up so that the action potential really does kind of jump from one spot to the next. And this is called saltatory conduction because that's what saltatory means. It means jumping like that. And in the
It's not related to salt.
what?
It's not related to salt.
I don't know where the word comes from exactly.
No, if the action, if like it's sodium that's.
Oh, no, it doesn't have anything to do with that.
Yeah.
It just happened because that's happening in the continuous or the saltatory conduction, whether it's going all the way down an unmyelinated axon or not. The sodium's still doing its job. So it's not about salt as in sodium chloride. I don't know where the word comes from exactly. But in this video, as they're talking about this, they're showing here in the node, there are voltage-gated sodium channels and voltage-gated potassium channels. So there are all the things that are necessary for an action potential to happen here. They're not in the membrane that's wrapped by the myelin. And so the myelin, let's jump on a little bit further, come on. So the myelin allows the action potential to happen here, and then happen here, and then happen here, and happen here. It moves along much quicker that way. Unmyelinated axons, because I have to do that continuous kind of conduction or propagation, will send signals very slowly at about, I want to say it's 2 meters per second or thereabout. And in myelinated axons, the signal travels much more quickly. It might be 50 to 100 meters per second because it's jumping from here to here to here to here. Think about, let me go back to this one here. In a myelinated axon, think about each little spot where you see the red band where the action potential, that's a node. And so it goes from one node to the next. And we don't have myelin in this picture, but it's as if there's myelin between the two spots there. So it's jumping from one spot to the next when it's myelinated. It travels much faster that way. Oops. So in the book, it's talking about propagation, and it defines continuous conduction and saltatory conduction there. And now you got me interested. I have to look up what the word saltatory, where that comes from actually. But so that's the material about the action potential. The next section, which is the last part of this chapter, is labeled communication between neurons. And what this is about is really about how the signal that's traveling along the action potential gets passed to the next cell and then causes that next cell to generate an action potential. So it starts here talking about graded potentials, which I actually already talked about, because that's what gets the action potential started. It talks about a few different types of graded potentials, generated potentials, receptor potentials, and postsynaptic potentials. Generator potentials and receptor potentials are found where the nerve endings, the dendrites, are interacting with some other stimulus. So when you're putting pressure on your skin, that's activating receptors in the dendrites. Receptors in the dermis. And if it's actually a nerve cell that's being activated, that's a generator potential. And if it's a specialized cell called a receptor cell, then it's a receptor potential. During the break, I was talking with Aaron a little bit about vision. And so photoreceptors are not actually neurons, they're a specialized cell. And so they'd be generating a receptor potential. That's just the difference between those two types. And then postsynaptic potentials are graded potentials that happen at synapses. It's called a postsynaptic potential because the target neuron is on the receiving side, the postsynaptic side. There's two types of postsynaptic potentials. There's excitatory and inhibitory. So in the second paragraph under types of potentials, the second paragraph is talking about postsynaptic potentials, and then it introduces excitatory or inhibitory postsynaptic potentials, which are abbreviated EPSP for excitatory postsynaptic potential and IPSP for inhibitory postsynaptic potential. And really what those are talking about are depolarizing and hyperpolarizing graded potentials. So a depolarizing graded potential that happens at a synapse is an excitatory postsynaptic potential. And a hyperpolarizing graded potential that happens at a synapse is an inhibitory postsynaptic potential. So just giving those things names. I talked a little bit about summation. Just if we add them together, we get more out of it. This is kind of a weird picture, which I hope makes the point. It didn't quite turn out the way I'd originally imagined it, but here we can measure membrane potential. So this yellow part of the line is at -70, meaning resting potential. If there's a depolarizing graded potential that'll bring the membrane potential up, but this one isn't enough to reach threshold. We have a second one that adds to that, we'll get up a little bit closer. We have a third one that's a hyperpolarizing potential, then it goes down. It actually took me a while to realize that we're back to yellow here. I have a little bit of color vision issues, so I didn't realize that this is a different color than the green. So I didn't realize that for a little while, but that's in fact what's happening. And the point here, and this is part of what makes this an even stranger picture than I thought, there's no postsynaptic potentials happening here. And so the membrane's going to try to get back to resting potential. So the yellow line's kind of falling down a little bit because nothing's happening. And then another excitatory or depolarizing graded potential comes along, and then a hyperpolarizing one, and then a depolarizing one. And finally, adding all of those up, positive or negative changes, we get to threshold. And once we hit threshold, we see summation. We see that axon hillock. I mean, we saw the action potential occurring. Now it's happening at the axon hillock, which is this part of the cell right here. In the video we started off with, when I was showing you the graded potentials that were all kind of converging on one part of the cell, where they were converging was the beginning of the axon, which is called the axon hillock, okay, right there. So in this picture, we have green synapses and purple synapses corresponding to the depolarizing or hyperpolarizing graded potentials. And depolarizing graded potentials at synapses are excitatory postsynaptic potentials, or EPSPs, and hyperpolarizing ones are inhibitory postsynaptic potentials, or IPSPs. So what this is showing is at each particular synapse, the same thing is going to happen. This is an excitatory synapse. This is an inhibitory synapse. Okay? And when all of these synapses are active, they can add up some positive, some negative results. And we eventually get to threshold there. Okay.
Yeah, so and like basically it's like kind of a competition between excitatory and inhibitory, like so, and like whichever one there's more of, if there's eventually gets to be more excitatory synapses, it polarizes, polarizes, but otherwise it like remain, it doesn't if you have like a certain amount more of one, either excitatory or inhibitory, like it determines whether you have a
Exponential or not.
yeah.
Right. So I can actually illustrate that a little bit. Okay. There's a part of your brain right now that's sending information to another part of your brain, which results in your conscious perception of what's going on around you. Okay. Actually, it's happening in a lot of different places in your brain. But in one of those places, you're paying attention, or your brain is paying attention to the tactile stimuli that are around you. Okay. Right now, well, no, I'm about to tell you something which is going to change how that's working. Okay. The clothes you're wearing are pressing on the skin in your body right now. Okay. Before I said that, you probably weren't thinking about your clothes. You just ignored them. But as soon as I said it, now you're thinking, oh yeah, I can feel my clothes. Okay. What I did is I caused a synapse to become active, which increased the likelihood of another neuron to be active that sends a signal on to the part of your brain that's consciously aware of what's touching your body. Okay. And so previously there might have been input coming in from your skin depicting the sensation of stuff touching your skin, but it never got to threshold. Okay. And then when I mentioned it, suddenly another part of your brain becomes active and you get a little boost and it brings things up. There might have even been parts of your brain that were suppressing that sensation of the clothes on your skin because it's not useful information, probably. It's nice to know that you're not running around naked, but you can probably assume that you're not running around naked from moment to moment in your life. So you suppress that feedback until something happens that you become aware of it, which doesn't necessarily have to be me mentioning it. It could just as easily be, you move your hand, you can feel the sleeve of your shirt moving or something like that. You become aware of the clothes again, and you reach threshold, and that information gets sent on to the part of your body that's consciously processing it. So does that kind of fit in with what you were talking about, Aaron? That idea? Yeah. So there are places, kind of like switches going on and off, based on how much excitatory or inhibitory activity is taking place, really determining if you do not hit threshold, and that's going to send.
So it's what I was trying to. It's like whether like it's always like one or the other, or whether it's or whether it's like a combination of the two, and whether it's always a combination of the two, and it's just a variance of which one is more most prevalent, and you kind of like said that like it's
Right. So, and in the example I'm giving, it's a rather complex situation. So there's going to be, sort of a complex input of both excitatory and inhibitory. If we look at simpler and simpler systems, it'll be a little bit more black and white. But sort of the furthest extension of what you're asking is, yeah, it kind of is an interplay between the two. So I want to go on to talk a little bit about what's going on in the synapses. So in the book, this little bullet list here is sort of defining the different things that we see at a synapse. And we've actually already talked about a synapse, which was the neuromuscular junction. So if we think about the neuromuscular junction, these things are all there, okay? The presynaptic element, that's the thing that's sending information into the synapse. That's the axon that's ending in a little bulb, the axon terminal, that's the synaptic end bulb. Okay? It contains neurotransmitters that are packaged in vesicles. That neurotransmitter for the neuromuscular junction was acetylcholine. Okay? There's a gap between the presynaptic element and its target, which we call the synaptic cleft. And that target is referred to as a postsynaptic element. That's the thing on the other side of the synaptic cleft, which contains receptor proteins. If there are not receptor proteins, the neurotransmitters can't bind to anything and nothing happens. So there has to be a receptor that's receiving this chemical signal of the neurotransmitters. And then there has to be some method to get rid of the neurotransmitters. As long as the neurotransmitters are present in the synaptic cleft, then the receptors will be activated and channels will open and ions will flow across the membrane. So we need to limit the signal. So there has to be something that gets rid of it. We can either just destroy the transmitter molecule, or we can take it back up into the presynaptic element for reuse, what we would call reuptake. And there's some variations of these, but basically either we completely destroy it or we clear it away and reuse it later on. This is a figure in this chapter that's very much like the figure in the muscle chapter, showing the components and talking about how when the action potential gets to the synaptic end bulb, it causes neurotransmitter release, which will then diffuse across and bind to its receptor. Now this next section, I think it's an important thing to talk about, but I think it's sometimes kind of confuses students a little bit. When we're talking about what's going on at a synapse, we want to think about the neurotransmitter that's involved. And so we talk about neurotransmitters as being part of a system. And the system is going to include not just the neurotransmitter, but also its receptors and some other features too. I want to keep this kind of simple, so I just want to concentrate on the neurotransmitter and its receptors. There's two types of receptors. I just want to mention this because we're going to talk about receptors in a little bit. Some of them, like what we've already talked about, are ligand-gated ion channels. Okay, so the neurotransmitter, like acetylcholine, binds to the receptor on the external side of the membrane, and that causes an ion channel to open and ions move in. That's a direct activation, or a direct action type of receptor. The other type is when it's indirect. The receptor protein is what the neurotransmitter binds to, and it's connected to some other proteins on the inner side of the cell. These things that look kind of like, I don't know, a Wi-Fi signal or whatever, is just representing that there's a change in the receptor protein that affects this thing on the inside surface. And when that changes, it causes metabolic changes or chemical changes inside the cell, which then propagate or influence other things inside the cell, like this ion channel here. So the direct one is an ion channel that opens because it binds to something. The indirect example is an ion channel that opens because some chemical signals got started because some proteins attached to the receptor are activated when the neurotransmitter binds to it. In these pictures, we're just showing one ion channel activated when something binds to it, or a second ion channel activated when more stuff binds to it. Here, we're showing one ion channel activated when something binds to this receptor protein. In actuality, the second picture down here, we could have multiple ion channels that are all activated at the same time. Okay. So the neurotransmitter binding to this type of receptor can actually open multiple ion channels. Whereas here, since it's binding to the ion channel, it's only opening that one ion channel. We can get a bigger response with this second type. The problem is that it takes a little bit longer for it to happen. Okay, all of the steps involved to get to opening the channel take milliseconds, whereas the binding that opens this channel here is practically instantaneous, probably measured in nanoseconds or something like that. So there's a trade-off between the speed of reaction and the strength of reaction between these two types. And I just want to mention those. In the book, I define them a little bit better. There's the ionotropic receptor, which is the direct one, and the metabotropic, which is the one that causes metabolic changes inside the cell. This table here is illustrating a few examples of neurotransmitter systems. Now, when we talk about a neurotransmitter system, the system has a name, and that name is based on the neurotransmitter that's in play. And in retrospect, I didn't quite label this table the way I should have. At the top, it says, in bold, system, and then it says cholinergic amino acids, biogenic amines, and neuropeptides. Actually, cholinergic is the only system name there because it's a unique one. There's actually 3 different systems represented as in the amino acid column and 3 different ones represented in the biogenic amines column. But those 2 columns are talking about different classes of neurotransmitters. And I'll give you the system name as we talk about it. Now, the cholinergic system, which is the only one in this sort of representative example, is based on acetylcholine. So hopefully you can see that the word cholinergic comes from choline, which is part of the neurotransmitter name, and then ergic at the end comes from the root of the word energy. So this is describing what's activated by acetylcholine. Now, there are two cholinergic receptors. They're called the nicotinic and the muscarinic receptors. The names come from the fact that we can use a artificial stimulant, or I shouldn't say stimulant, an artificial agent to bind to the receptor and cause a change. Okay, and that's what the two types are named after. The nicotinic receptor is named after the fact that nicotine can bind to it. It's kind of unfortunate that it's nicotine because everybody's heard of nicotine, you know, that it's a component in cigarettes and tobacco, which is just coincidental at this point. The natural effect that nicotine has on the nervous system is something that we'll talk about in A&P2, where it has some relevance. At this point, we're not going to get bogged down in that. So it's just a drug that activates this type of receptor. And then the other one is activated by a drug that's called muscarine. And that's kind of nice because you've probably not heard of muscarine, so it's not making you think of something that's going to confuse you about things. But the idea here is there's two types of receptors, and they're classified because of an artificial agent that can be used to activate them under unusual circumstances. But really what we're talking about is that these are two receptors that acetylcholine can activate. Okay. Where they're found in the body varies because of how they work in the body. Okay. The next part of the next row in the table there is talking about elimination, how we get rid of that. I'm going to skip over that because I don't want to spend too much time talking about it, but it does mention what happens to all of these things at the synapse. What's important is the postsynaptic effect. The nicotinic receptor always causes depolarization, okay? Always causes excitatory postsynaptic potentials. And that's the acetylcholine receptor we've already looked at. Okay? That's the one that's an ion channel that when acetylcholine binds to it, it allows sodium to cross the membrane and therefore depolarize it. That's what we've been talking about. There are actually a few different types of muscarinic receptors. Some of them cause depolarization, some of them cause hyperpolarization. They're all those second type of receptors that I showed you before, those metabotropic ones that control ion channels at a distance. And so depending on the particular subtype, the ion channels it opens can vary. If it's an ion channel that allows positive ions into the cell, then it's going to cause depolarization. If it's an ion channel that allows negative ions into the cell, then it's going to cause hyperpolarization. Or it could be an ion channel that allows positive ions out of the cell. What's going to happen to the membrane potential when positive ions leave the cell?
Negative?
Yeah. So you take the positive out of the cell, becomes negative. The same thing as if negative ions entered the cell. Okay, so negative ions entering or positive ions leaving both cause hyperpolarization. And then positive ions entering causes depolarization. That might make you think, okay, maybe negative ions could leave the cell also. In fact, that doesn't happen. There are no negative ions that are higher concentrated inside and outside of the cell that can pass through an ion channel. The only ones that are the proteins and other things that I mentioned earlier, which is what makes the inside of the cell actually more negative than outside. But they can't leave the cell. So you can't have negative ions leaving a cell. It's only positive ions entering, positive ions leaving, or negative ions entering. One of those causes depolarization, the other two cause hyperpolarization. There's two different ways that can happen. And if it's a positive ion leaving the cell, it's potassium. If it's a positive ion entering the cell, it's sodium or chloride or something like that. And then negative ions that enter the cell is usually chloride ions. So muscarinic receptors control different types of ion channels. And depending on which type of receptor we're talking about, which subtype, It's either going to cause depolarization or hyperpolarization in those different ways I just mentioned. Now, the next column under amino acids is actually 3 different systems. And again, the system name comes from part of the name of the neurotransmitter with the ending ergic. And the 3 amino acids that we're talking about here are glutamate, glycine, and GABA. So glutamate is a system unto itself, which would be called the glutamatergic system. Glycine is a system unto itself, and it's called the glycinergic system. And then GABA is an abbreviation for a longer term that I'm not going to bother introducing because we just call it GABA. And in one of the more awkward word constructions, the system that involves the neurotransmitter GABA is called the GABAergic system, which is just kind of a weird way of putting it. The receptors that we see here are just named for what binds to them. So we have glutamate receptors, glycine receptors, and GABA receptors. There are different types of glutamate receptors. There are different types of GABA receptors. I don't remember if there's more than one type of glycine receptor, but it doesn't really matter. What I'm saying here is that all glutamate receptors do the same thing. All glycine receptors do the same thing. All GABA receptors do the same thing, which is what it says at the bottom under postsynaptic effect. Glutamate receptors cause depolarization. Glycine and GABA receptors both cause hyperpolarization. You'll sometimes hear the amino acids referred to as excitatory or inhibitory amino acids. Glutamate is an excitatory amino acid because all of its receptors cause excitatory postsynaptic potentials. GABA or glycine are inhibitory amino acids because they always cause inhibitory postsynaptic potentials. And then the next column here are biogenic amines, which the examples I have here are serotonin, dopamine, and norepinephrine. Biogenic amine is saying that these are neurotransmitters that are made in the body. That's what biogenic means. And they have an amino group attached to them, amines. They're not amino acids. They no longer have anything making them organic acids, but they were made from organic acids. Serotonin is made from the amino acid tryptophan. Dopamine and norepinephrine are made from the amino acid tyrosine. How that happens isn't terribly important right now, just that's where they come from, chemically. The receptors for serotonin are referred to as 5-HT receptors because 5-HT is an abbreviation for the chemical structure of serotonin. There are subtypes of 5-HT receptors, but we're not talking about them because they all do the same thing. They're all hyperpolarizing. They all cause inhibitory postsynaptic potentials. Dopamine receptors are classified as either D1 or D2 receptors. I often get these backwards, so I'm not going to actually try to say which is which, but one of them causes excitation, the other one causes inhibition. And which is which isn't relevant right now. It will come up later in the course, and I'll try to say it correctly when it's important, but there are those two types. So dopamine can cause either excitation or inhibition. Okay. And then norepinephrine is a chemical that's also known as noradrenaline. Okay. And then in parentheses there I have the word epinephrine, which is also known as adrenaline. Epinephrine and adrenaline are just the Greek and Latin versions of that name. And then the nor on the beginning of the word is just a chemical difference between those. I have epinephrine in parentheses there because epinephrine isn't really a neurotransmitter. It's a hormone, but it's very closely related to the neurotransmitter norepinephrine. In fact, the only difference is epinephrine has like an extra carbon added to it. That makes it more.
Does that epinephrine cause like release of norepinephrine?
No. So at a cell will release these signaling chemicals. Some cells release epinephrine, some release norepinephrine. The ones that release epinephrine are always endocrine glands.
Yeah.
The ones that release norepinephrine, sometimes they're endocrine glands, but more often they're neurons.
Yeah, but I guess I was wondering is like whether epinephrine, like the hormone, when if it binds to would epinephrine bind to a neuron and then cause like that to then release norepinephrine.
I'm not aware of any situation where that's the case.
Yeah,
And it would probably not, because that would be rather inefficient. Epinephrine and norepinephrine actually bind to the same receptors. So there certainly could be situations where that's true, but for the simplest explanations, it would be redundant. Because if epinephrine bound to something that caused norepinephrine release, then that norepinephrine would cause some change, which the epinephrine actually would have done to begin with.
And they're basically just like in different places in the body. And
Yeah, and actually in the AMP2, we'll talk a little bit more in depth about that particular pair of chemicals because they're rather important. So you'll learn more about that in AMP2. But like I was saying, epinephrine is just another way of saying adrenaline. Norepinephrine is also known as noradrenaline. The adrene in adrenaline is referring to the gland that's on top of the kidneys. Ad means on top of. Rene is the Latin root that means kidney. Epinephrine is the Greek way of saying the same thing. Okay. So epinephrine is the chemical that's made by the adrenal gland. That's essentially what it is. The reason why there's two names for it is this weird little quirk in U.S. trademark law, which I don't want to get into here, but that's why there's two names for it. The system that norepinephrine belongs to is called the adrenergic system. And there are two receptors called the alpha and beta adrenergic receptors, or sometimes just simply the alpha and beta receptors. If you've ever heard of a beta blocker, that's a drug that blocks the beta adrenergic receptor. And we'll talk about that in AMP2, because those sorts of drugs have to do with cardiovascular function. Now, a second ago I was saying the D1 and D2 receptors, one of them is excitatory, one of them is inhibitory. The alpha and beta receptors, it's not that one is one way and the other is the other way, but there are multiple types of alpha and multiple types of beta receptors. Some of them are excitatory, some of them are inhibitory. Now, at the bottom of the biogenic amine column there, it says essentially that depolarization or hyperpolarization depends on the specific receptor. For example, D1 receptors, oh, here I have it written out, cause depolarization, D2 receptors cause hyperpolarization. You could also say some alpha and some beta receptors cause depolarization, some alpha and some beta receptors cause hyperpolarization. The serotonin receptors all do the same thing, which is hyperpolarization, which it unfortunately doesn't say there. In retrospect, I wish I put that in there, but it doesn't. From this, what I want you to pay attention to is the neurotransmitters, what their receptors are, basic names of receptors, and what those receptors can do. So the receptors for acetylcholine, the nicotinic receptor always causes depolarization. The muscarinic receptor could cause one or the other, depending on the particular subtype of receptor. Glutamate receptors always cause Glutamate receptors always cause depolarization. Glycine and GABA receptors and serotonin receptors always cause hyperpolarization. Dopamine receptors, one type causes excitation, the other type causes inhibition or depolarization, hyperpolarization, same way of saying that. And then the alpha and beta receptors, some of them cause depolarization, some of them cause hyperpolarization. So some of these are neurotransmitters and their receptors that always cause the same change. And some of them, it depends on the particular receptor what results can come of it. Okay. The last column here that says neuropeptides, I put this in there just to talk about other types of substances that can be active at synapses. I don't really want to talk about them. I don't ever get into that in this course. So understand that such things exist, but I'm not going to ask you about these. I want to just concentrate on the cholinergic, glutamatergic, glycinergic, GABAergic, serotonergic, dopaminergic, and adrenergic systems. Okay, so these three columns here. Just there's several different systems listed there. What I think that's 7 total. 1, 2, 3, 4. Yeah, 7 total. Some of them do only one thing. Some of them, depending on the receptor, do multiple things. So that's kind of laid out down here. It does not actually mention specifically that serotonin, like glycine and GABA, always causes hyperpolarization. But I want to point that out there because that's true. So that gets to the end of that section there, which gets us to the end of the chapter. Now, the next chapter is about the anatomy of the nervous system. And then following that is the somatic nervous system, which is really about the functional parts of the nervous system that we're concerned with this semester. So next week, we're going to be looking at the anatomy of the nervous system and the somatic nervous system together. The assignments in next week's folder that are called the nervous system, part 1, 2, 3, 4, are based on these 2 chapters here. We will not be doing the autonomic nervous system this semester. That's an A&P2 thing, which is where we're going to talk about nicotine and epinephrine and norepinephrine in more detail. And then the week after next, we're going to be considering this last chapter on the nervous system, which is the neurological exam. There's no assignments associated with that chapter. There was a learning pod, but of course we're not doing learning pods anymore. There are no chapter assignments for it, but we'll be considering what's clinically important about the nervous system there, which will actually tie into what we're doing next week, which is about the anatomy and somatic nervous system functions. So we'll have sort of the traditional look at the anatomy and physiology of the nervous system next week. And then the following week, we'll consider what we learn through neurology about what's going on there. And we'll come back to that. So that's what we have left in the semester. Now, we certainly have more time today, and I could go on and start talking about the anatomy of the nervous system. But I think the material we've covered today is kind of dense, and I want to stop here so that you have time to process that over the next week, and then we'll get into other material next week. And next week, we're going to be looking at models of the brain like I have on the card here. My night class tonight is up to that point, so they're going to be doing that, so I have not. But that's what we're going to be looking at next week to go along with the anatomy of the nervous system stuff that we're doing. Okay. And again, that includes some of the material that's on the lab quiz in this week's folder, but I pushed the due date back till after next week because you won't get that information until next week anyways. So, so that's all I have for today. And I'll see everybody next week.
Yes.